Hello everyone, I would like to introduce Elizabeth Mahone, and the title of her talk is going to be "The Proof is in the Partially Digested Plants, Reconstructing Chrysopus Cremopinidae, Chrysomelidae, Phylogeny and Host Classifications through Gut DNA Extractions."
Thank you. Can everybody hear? Okay. So I'll be talking about the start of my research on this plant herbivore system, and really just about the method I'm trying to develop to look at this. So almost all insects have some kind of association with plants, but the majority of them are actually host plant specialists. And these are some really well-studied examples of those types of associations. It's been hypothesized that these tight evolutionary relationships are mediated in large part by the plant's secondary chemistry, and that in turn might be the cause of the great phenotypic and species diversity in both plants and Phytopagus insects. So by one estimate, green plants together with Phytopagus insects make up nearly 50% of the estimated biodiversity on Earth. And one of these groups of highly specialized insects are the Chrysopus beetles. They're in the leaf beetle family, Chrysomelidae, and the Eumolpinae subfamily. And there are 10 currently recognized species, 2 of which are North American. Those are the ones pictured on this slide. And there are about 8 in Eurasia. There are 2 species of the segregate genus, Chrysocharis, in Asia also, and a closely related sister genus, Platycornis, in Asia and Africa. So in North America, we know that these 2 species have host plants in the Aposemaceae family that produce cardenolides, which are cardiotoxic steroids that are unpalatable to most animals. But these beetles have a physiological insensitivity that allows them to eat cardenolides. There are a few other cases of this. Didaeus plaxipus, monarch butterflies are another good example. But these beetles, like them, can actually sequester the cardenolides and possibly use them in their own defense. And another well-studied species in this group is Chrysopus asclepiadaeus in Eurasia. And they also feed in plants in the Aposemaceae, but their host plants are Vincetoxicum, which don't produce cardenolides. And beetles have been shown not to have that physiological insensitivity to cardenolides. So, like I said, we know right now that these 2 North American species are insensitive. The Eurasian species is not. And then the bulk of the group is actually in Asia, and their cardenolide use and possible tolerance or intolerance hasn't been studied yet. So similarly, we know the host plants of the North American species are Opossum and Asclepius, making cardenolides. The Eurasian species, Vincetoxicum, which doesn't have them. And then most of the host plants are actually unknown for the Asian species. Although there are a couple of reports of a Chrysocharis species feeding on one plant that doesn't have cardenolides and possibly an opossum, but it's unclear which one is described there. So given all these big gaps in post-association information and the taxonomic literature for Chrysopus, my first goal with this project was to actually construct a phylogeny of Chrysopus sensolata, which hasn't been done, and identify the rest of the host plants in the group and map them to that phylogeny, and then use those data to start reconstructing the historical biogeography of Chrysopus and Opossum. Opossum in particular, because there aren't many genera in the Aposemaceae family that have this North America, Eurasia distribution like Chrysopus does, but this is one of the few genera that does. So again, there's not much information on these associations for the Asian species, and what's there can be unreliable. So some common problems have to do with host plant misidentification in the field. Taxonomic confusion, so I referred to Vincetoxicum and Cynancum a couple slides ago. And Vincetoxicum, some species used to be included in Cynancum, but the distinction is actually really important to a beetle because Vincetoxicum don't have cardenolides, but some Cynancum species do. And then when you don't keep a host plant voucher, when you're collecting beetles, that makes looking at these interactions later more difficult. So just for an example of why I think this is a real problem, here's our more prevalent North American species, Chrysochus auratus. And these, like you can see, there are two reported host plants next to each other. And you can see how difficult they might be to tell apart if you're not really familiar with this group. So for these associations that aren't really reported in the literature yet and might be difficult for somebody like me working in Philadelphia to go out and study, I thought the next best thing would be to actually directly ask specimens in museums. So I started trying to develop a way of extracting host plant DNA from museum beetle specimens while still preserving their use for future morphological study, especially for the less well represented species. So a few different, so different groups out there have done similar things, and I was looking at approaches that might be appropriate for what I want to do. By far, the most common methods for doing something like this are specimen destructive. So obviously not useful for, if you want to preserve the specimens for the future. But there's a really nice recent example by Garcia Robledo and his colleagues who collected chrysomelas and then ground them and got the host plant and beetle DNA out of that. I also looked at dissecting out the guts and talked to Jessica Ware at Rutgers, who's doing something kind of similar with cockroaches mainly. But this seems to work a lot better with those big flexible cockroaches than small dry beetles. And the last method that I looked at was putting specimens into an extraction buffer and then removing them and extracting DNA from the buffer. So a few people have done this mainly for looking at the insect DNA, but also the bottom two references have done this with newly collected specimens and got some host plant DNA. But I wanted to point out this Gilbert paper from 2007. They were just looking at beetle DNA, but they actually extracted it from specimens up to 94 years old. So I thought that seemed really promising, and you can see the pictures of, those are the after pictures of this method. So I thought, it'd be great if we could try that with, old specimens from museums to get the host plant DNA too. So basically, my methods for DNA extraction, I've tried dissecting out guts and also tried this extraction buffer, sometimes puncturing the thorax of the specimens if they don't already have a pinhole. And then I've used some really commonly used DNA extraction kits. And for amplifying plant DNA, I've focused on the chloroplast tRNL intron, which is used in DNA barcoding commonly in plants. And for beetle DNA, I've focused on the sodium-potassium pump region, which has to do with the cardenolide tolerance and intolerance, and also mitochondrial COX-1 region for insects. So this is just showing one of the first tests where I dissected out guts and used these other immersion methods with newly collected Chrysochus auratus. So this is the North American, the eastern North American species that we know they feed on opossum. So it was amplifying DNA of the same size for tRNL. And then I tried this with some specimens from the Academy of Natural Sciences that were older, dried specimens. Did not work so well. We got right size products for a couple more recent specimens. And so this is just showing the circles on this chart are dried specimens from the museum, and I had many more ethanol reserve specimens. So unsurprisingly, it works much better in the newer specimens. And then confirmed that plant DNA being amplified was a opossum for these Chrysochus auratus specimens. So these are just three extractions from 3 different beetles that matched up with opossum and blast searches. So started trying to expand this to the taxa that we actually care about because we don't know their host plants yet, in Chrysochus and Platycornis. And so far tried 54 specimens from 17 different species in those 3 genera, ranging from 5 to 109 years old, and these are all dried specimens from different museums or donations. And they were collected on 3 different continents, and I've mainly tried those immersion methods. So the very first attempts amplifying DNA from these looked really promising. We were actually getting products from a lot of these older museum specimens of these different species. But of course, there was a problem. The majority of the species, when we sequenced them, were pine, oak, juniper species. So again, we expect most of the host plants will probably be in the Opossinaceae. And we did get a few that these 3 were from beetles that we already know their hosts that were going to be correct. But this DNA was all extracted in March and April in Philadelphia, and since we had a few species that we knew their host plant that were also getting pine species as the host, the more likely answer here is that we have pollen contamination in a lot of samples. So this has been pretty difficult to get around, actually. But we've basically been ramping up our sterilization methods for DNA extractions in PCR, trying to make that more rigorous. And it seems like it's starting to work now. Contamination seems to have disappeared at this point. Although the only way to really get around this might be to, with all these sterilization procedures, do this in the winter. So in just some very recent PCRs, we don't have contamination, but we also don't have PCR products, unfortunately, from those museum specimens. So obviously, the way to get around, or a good solution would just be to collect beetles in Asia, see what they're eating at the time, and then do more extractions to confirm maybe other possible hosts. And so I'm going to be doing that in China in a few weeks, trying to, the majority of the Asian species live in parts of China, so I'm going to be making those collections. But I still think this technique, should have some use, especially for species that are rare or, just when it's difficult to get to the places. A lot of these are actually in Russia and Kazakhstan, not the easiest places always to visit. So right now I'm still working on optimizing the extraction methods for museum specimens and planning to make collections for them. So need to thank a couple students, Yasha and Christina, have started helping us on that. And all these people, for specimens they donated